Welcome to this presentation on services offered by MutaMouse, the transgenic core facility of the University of Leuven. At MutaMouse, we aim at supporting and realizing your genome editing projects, both in vitro and in vivo, as well as providing you with embryo services. Those services comprise the generation of mutant mice, the production of chimeric embryos and mice, long-term strain preservation as well as strain recovery, embryo rederivation to introduce newly imported strains into the KU Leuven animalium, and support with existing mouse strains, such as the rescue of mouse strains. Our team is led by Professor Adrian Liston and has expertise as well as strong technical support in transgenic and several additional mouse services. With the next slide, we would like to introduce our genome editing services using CRISPR-Cas and provide you with an overview how mouse genome manipulation can be useful to your research. Currently, undoubtedly the most common application of CRISPR-Cas is the generation of knockout alleles. As illustrated on the right, the preference of the cells to repair double-strand breaks introduced by the Cas9 endonuclease is an imperfect repair mechanism. The imperfect repair results in insertions or deletions of nucleotides, which, if in an exon, can result in a frameshift. Consequently, a premature stop codon will appear downstream of the mutation and result in a knockout of the gene of interest. You can bring your own tested CRISPRs, or we can help you find appropriate CRISPRs. For knockouts in cell lines, we offer the possibility to bulk sort your cells to enrich after transfection, and to single cell sort to generate individual clones. To generate knockout mice, complexes of CRISPR RNA and Cas9 protein, so-called ribonucleoproteins, are injected into early embryos. Similarly, large deletions can be achieved by the use of 2 CRISPR binding sites flanking the to-be-deleted region. The transgenic founder mice will be the basis for the desired new knockout strain. Other studies may require the modification of a gene by exchange or addition of sequence. Examples are the insertion of point mutations that resemble human disease-causing polymorphisms, or the tagging of a protein of interest with an epitope, such as flag tag, for imaging or biochemical purposes. The gene of interest can be turned into a conditional allele to allow temporal, spatial, or cell type-specific knockouts. Most commonly, LOXP sites will be inserted, which are recognized by Cre recombinase. Cleavage will delete the LOXP-flanked sequence. Alternatively, the gene of interest can be linked to a fluorescent reporter such as GFP or a recombinase such as Cre. In the case of GFP, active transcription is visualized. In the case of Cre, live and past activity of the gene can be followed. Knock-in experiments require, in addition to a functional CRISPR, also a donor DNA that serves as a template for homologous recombination. Our team can help you with the design as well as the cloning of your donor DNA. Complex knock-ins will be performed in mouse embryonic stem cells. Those cells will subsequently be injected to generate chimeric mice. For smaller knock-ins, such as epitopes or point mutations, it can be possible to achieve knock-in by direct pronuclear injection. However, success rates can vary with CRISPR activity and locus accessibility. An exception are the so-called safe harbor loci, such as ROSA26. These loci are constitutively open and their modification does not result in any unwanted phenotype. Therefore, the ROSA26 locus is one of the model loci to insert transgenic overexpression cassettes or to insert Cre-sensitive reporter alleles. We can provide support designing a ROSA26 allele for a specific purpose. Depending on your donor design, ES cells are modified and subsequently injected, or the donor can be co-injected with ribonucleoproteins into early embryos to obtain founder mice. Pricing is given for individual parts of the genome project, such as CRISPR editing or ES cell manipulation, or for full services such as all-inclusive generation of knockout mice. These fees are paid in advance and the project is initiated upon receipt of payment. Even though we work with 3 different endonucleases for genome editing, it can happen that no working CRISPR can be found in the region of interest. In this case, an efficiency report is provided and 50% of the knockout full service fee is refunded. Knockout or knock-in full service guarantees the delivery of 5 founder mice in case of knockout generation or 5 chimeric mice in case of knock-in generation. If multiple injection rounds are required, we offer a 50% discount from the second round onwards. As for publications, we request acknowledgement if the mice or cell lines have been designed by the end user but made by MutaMouse, and we request a co-authorship if the mice or cell lines have been designed and made by us. Hopefully this part of the presentation gave you an overview of our genome editing services. At the end of this presentation you will find our contact details would you like to get in touch with us and arrange a meeting. I'm now handing over to my colleague Lutgarde, who will introduce our mouse generation, strain preservation and embryo rederivation services.
As explained before by Susan, we can generate mice or embryos by different methods. If you have a more complex project, like integration of large pieces of DNA or conditional knockouts, we will perform the genome engineering in embryonic stem cells. And after we identify the correct clone, we will inject those cells into a blastocyst, or we will aggregate the cells with a morula embryo. After germline transmission, your new strain is established. Besides this, we can also inject embryonic stem cells you obtained from mutant mouse consortiums like COMP. Per project, we will inject your clone in 2 independent sessions, and you can expect to obtain 5 or 10 pups. And depending on the totipotency of your stem cell, we will generate in these experiments 2 to 10 chimeric animals. We can also perform all mouse generation methods to address tailor-based questions. In case your project involves a simple knockout by introducing a reading shift or a stop codon, a chromosomal deletion or an integration in the ROSA loci, we can provide zygote injections. We can either inject DNA, RNA or ribonuclein protein complexes into the cytoplasm or into the male pronucleus of a zygote. This zygote can be wild type or already mutant if you prefer this. Pronuclear injection is the method for your project if homologous recombination or DNA integration is involved. The generation of a simple knockout or a chromosomal deletion with the CRISPR-Cas method can be done by a simple cytoplasmic injection. From one zygote session, you can expect to have 10 to 5 pups born, potentially containing indels. In our hands, we see that about 7 to 80% of the pups contain indels. Contact us in case you have a tailor-based question. Besides the creation of new animals, we offer methods inherent related to mouse work, like cryopreservation, in vitro fertilization, and rederivation. For those methods, we are collaborating and we are sponsored by the KU Leuven Animal Facility. Cryopreservation is a safeguard to your research from infections, from genetic drift and loss of transgenes, or from breeding problems. Or in case you want to consider to stop active breeding for a while. While it can be problematic to transport live animals abroad, for example because they are infectious, sending and receiving frozen samples is always possible. If you consider to cryopreserve your strain, do this when your strain is still in active breeding, as we need fertile animals. To be completely on the safe side, we store the samples separated over 2 tanks in 2 separated locations. You can save money by cryopreserving your mice. See the diagram depicted here for a virtual case over 5 years. If you would not make use of your strain for 1.5 years, You save not only on your budget, but you will also contribute to the 3R principle. We offer two methods. In most cases, sperm freezing is the cheapest and technically easiest method. We ask to provide us with 5 proven fertile young males, aging 3 to 8 months. Ideally, those males are homozygous for the mutation. Our success rate of in vitro fertilization with frozen sperm is 94%. But in case you will stop your colony, we can also offer a quality control to live-born pups. We can also cryopreserve embryos. In this way, the genetic background of a complex strain can be maintained. As the number of collected embryos is depending on the strain performance, we conduct sessions with maximum 10 females. We collect and freeze all embryos after ovulation of young females that age 4 to 6 weeks. As a safe guide, we recommend to freeze 250 embryos from a homozygous strain and 500 from a heterozygous strain. On average, we collect 10 embryos per female. Also, we can always assist you with the revitalization of your own strain or from a strain of one of your collaborators. The hygienic status of your animals can affect your experimental results, but also your collaborations. If you need to upgrade the hygienic status of your animals, we can offer you to rederive your strain. The cheapest and technically easiest method is C-section. This service is offered by the Animal Facility. You can contact Bart Hermans in case you need more information. Some pathogens can cross the placenta. In those cases, embryo rederivation is recommended. We can also assist you together with collaboration we have with Charles River in speed back crossing in case you need big cohorts of your strain or to determine your genetic background of the strain. Please contact us. Last but not least, thanks for your attention and please feel free to contact us with your questions related to genome engineering of cell lines and mice and our mice services.